Sorry about that. So today I was planning on lecturing the translation portion more than the transcription portion. But when I just asked you on a scale of 1 to 5, I didn't feel like a lot of you seemed confident in the transcription. So I think I maybe should go through the transcription piece. So I put both lectures in today, transcription and translation. I'm going to save the translation. I'll put translation off to Wednesday, and I'll do transcription today. Or after this, you're going to do a POGL. The POGL is over translation. I had one person communicate with me this weekend and say, I feel pretty good about transcription, but not translation. Could you go over that? So that's why I made the translation instead of the transcription. So we're going to go, first of all, central dogma. You guys described central dogma in your notebook. All right, when we talk about central dogma, we're really talking about the flow of the genetic information through the whole cell process. So it's DNA making more DNA. It's DNA then making RNA. It's RNA then making proteins. So it's how do we get the code that's in the DNA into a protein ultimately, right? So so it happens in 2 steps: transcription, which is transcribing, and then translation, which is making sense. So when you see central dogma in an AP test format, you're going to think DNA to RNA to proteins, right? You should be able to talk about that entire process. Right now, we're talking about DNA and the enzymes involved, right? So I should be able to talk about the job of each of those enzymes as I explain DNA replication. I should be able to talk about the enzymes involved with creating mRNA. I should be able to talk about the steps that happen to modify mRNA to isolate a single gene or to modify that single gene to make the various proteins that we have, okay? Then I should be able to talk about the steps of translation and how those amino acids eventually become proteins. Okay, so you know that DNA codes for RNA and RNA codes for genes. So you want to make sure you're recognizing that it's coding for a gene and the gene is the code for a protein. Okay, just being really petty there. Proteins determine your phenotype. Remember, your phenotype is how something is expressed. And we're used to just looking at a phenotype as to what color hair you have, how tall you are, the shade of your skin. But your phenotype is also things that you can't see, like your blood type, whether you're A or B or O or AB, which is something you could see in a lab, whether you make an enzyme that allows you to break down lactose, any genetic disorders, like a lot of them are related to enzymes that need to do things. And if that enzyme, which is a protein, doesn't function, it leads to disorder. So phenotypes are not just the physical that you do see, but they're like the physicality or chemical reaction that is inside your bodies as well. So you want to kind of be a little bit broader in your thoughts there. Okay, and then we know that proteins serve as building blocks for cells and enzymes. We use them to make pretty much everything in our body. So how do we know that genes code for proteins? This comes from some famous, did you guys read about Tatum and Beadle in this section? So I'm going to talk a little bit more on that next slide. So how we came to the conclusion that genes code for proteins or genes code for enzymes that become proteins is through the studies of metabolic disorders. So just like I was just talking about how if you didn't have a certain enzyme, lactase, you couldn't break down lactose. But finding the connection between the two is what allowed scientists to say that genes code for enzymes. There's a gene per enzyme. So in this metabolic pathway, you have 1, 2, 3, 4 different enzymes that are involved. And we now know that like this one enzyme isn't responsible for the entire pathway, right? Every enzyme is involved with making an intermediate along the way. So each of those proteins that it makes would be referring to its phenotype, okay? So a lot of times we're lacking an enzyme that's needed to break down things like Tay-Sachs needs to break down lipids in the brain. And if you lack that enzyme, you can't break down the enzymes in the brain. PKU, that's, I think I showed you guys the footprint of the babies, so they can't break down phenylalanine. And then albinism, which doesn't manufacture melanin, which is pigmentation. So those are all due to a single gene enzyme malfunction. So this is the Tatum and Beadle experiment that came to the conclusion that one gene, one enzyme, so one gene makes one enzyme. So they worked with these fungal spores, which reproduce asexually. You guys did the ascus of Sordaria, right? So that should look familiar to you. And what they did was they used X-rays of ultraviolet radiation to create various mutations. They grew them in a medium. And so this is the positive control. So then what they did was one by one, they destroyed different enzymes. They were able to get rid of specific or create specific mutations. So the negative control is you're expecting not to see anything happening, and then the positive control, you're looking for differences. And then so as they messed with each of these different amino acids, they were able to isolate only this one amino acid led to the mutation, basically. And so that led them to the one gene, one protein, because they were knocking out one gene at a time. So, so two-step process, transcription, translation. We'll do transcription today, which is transcribing, which literally is just rewriting. Translation is the building of the protein, and then the transcription is rewriting the DNA into a form that can leave the nucleus. So DNA, remember, is a double-stranded helix, which is a little bit larger than a single-stranded piece of genetic code. Remember, the nuclear envelope has several pores, so perhaps this double-stranded nucleus is too big to fit through the pores. Plus, it would be pretty inefficient since our DNA carries all of our genes, and we don't need to code all of our genes. Like, we don't need to make all of those proteins all the time. We only need certain proteins at certain times. So to take this recipe book, if you will, and copy one single recipe would be much more efficient than taking the whole book, right? So we're going to transcribe the single gene that we need, and then that is small enough to fit through the pores of the nuclear envelope and go out into the cytoplasm where it finds the ribosome. So this is the ribosome. You should notice the double piece structure, basically. These might be free floating in the cytoplasm. It might also be attached to endoplasmic reticulum. So we talked about rough ER. If this was bacteria, the ribosome itself would attach to the RNA while the RNA is being transcribed on the DNA. So it's much simpler than DNA. We're making comparison, right? It's single-stranded. It uses ribose. We already looked at that last week. We looked at the fact that it's using uracil instead of thymine. Adenine is still there. So a lot of times kids make mistakes when they want to use adenine, they use uracil. So you only use uracil when you want to use thymine, okay? So we have several different types. 3 are very familiar to you, hopefully, MR and tRNAs. siRNA should be one that is new to you, the silencing or small interfering RNA. So messenger RNA, we want to know what each of these do, right? And you want to be able to talk about their process. So it's forming just the RNA from the complementary DNA, from the coding strand. It travels from the nucleus to the ribosome, where it will direct protein synthesis, and this is what that looks like. This is the 2 subunits of the ribosome. This would be your mRNA aligning with the ribosome, and this is your polypeptide growing strand. Each of those circles are different amino acids. So ribosomal RNA is embedded within this ribosome, and it helps this messenger align in the proper location so that it can connects to the A and P and E site of the ribosome. So transfer RNA, what is it transferring? It's transferring amino acids. So the codon on the mRNA, 3-letter right here, codon, codes for a specific amino acid. The anticodon brings that particular amino acid to the ribosome. So the tRNA has this traditional cloverleaf. A lot of times it's described as a cloverleaf structure. So this is all tRNA, and really the only piece that's important is that anticodon, those 3 letters, and it carries so those 3 letters bring a specific amino acid. So the one that should be new to you has to do with gene regulation, and it's small interfering RNA. It might also be called silencing RNA. So it is actually a double-stranded small segment of RNA that blocks blocks translation. So it interferes with the expression of specific genes, meaning it interferes with the translation of that gene, so it's not translated. So so you can see the double-stranded section here. So that's going to be like blocking a section of that RNA because maybe you don't need that protein anymore. Okay, so transcribing is taking the RNA or taking the DNA and making RNA. It does occur in the nucleus, so transcription, nucleus, translation, ribosome. You are going to use RNA polymerase. So we used DNA polymerase last week. It makes DNA. This one polymerizes RNA, meaning it makes long chains of nucleotides connected together in an RNA form. It will be complementary to the DNA, so you have a coding strand and a template strand. So this is the DNA. The DNA template strand, it's serving as a template, just like your cookie cutter serves as a template, right? This is the one that you're going to read. The coding strand is the code you want, right? So if the coding strand is complementary to the template strand, in order to get that code, I would have to make a new coding strand, basically, right? So you're going to transcribe the template, not the code. You're creating the code. So now this message can be read at the ribosome, okay? So we're going to call this pre-mRNA because it's not quite ready to go to the ribosome. So this happens in 3 steps. You'll have these same names for translation. You have initiation, elongation, and termination. Hopefully you can just recognize that's beginning, getting longer, and ending, right? So promoter regions. So RNA polymerase will bind to a promoter region. So that's the beginning. So it's a set of DNA nucleotides that the RNA polymerase would recognize and bind to. So that begins the transcribing. Sorry. Elongation is bringing in new nucleotides complementary to that template strand and adding those nucleotides. So you can see the messenger RNA forming over here. Termination, you get to a certain code, a specific sequence will initiate a release factor. And so then the mRNA will be released from the DNA, and now it's free floating, and it can begin the process of modifying in order to make it to the ribosome. Okay, so the promoter region we refer to as the Tata box. Here you can see a bunch of A's. So this is the sequence that are for eukaryotes that the promoter, that the polymerase is looking for. So it'll find this specific sequence that matches, you know, the shape of the promoter, and then that's where it will bind. So then polymerase can bind here, and then we begin our transcription with the help of several transcription factors, other proteins that are going to aid in the process. You have what's called an enhancer region. So in 9th grade, we wouldn't have talked about the enhancer region. It's basically a segment upstream from the promoter that is going to allow for a higher speed of replication or of transcription so that you can make more when you need more. So I thought I had a picture of that. The initiation, so the transcription factors, different proteins, see here, lots of different proteins are involved with attaching that polymerase. And just like you had the single binding proteins on the DNA replication, they're going to aid in holding that bubble open, basically, so that you can transcribe. So it's a suite of proteins. They bind to the DNA, and they aid in turning off or turning on a gene, because we don't need to transcribe all genes all the time. Okay? So you can click this maybe later. You can do it on your own. Okay, you can't. I'll fix that. But this is just transcribing, as in, if your code was ACG, what would the other side be? So if DNA was ACG, what would the mRNA be? A would bind to U, C would bind to G, and G would bind to C. So you would have UGC. That would be transcribing. Simple as that. Okay, many polymerase enzymes. Just like we had many polymerase enzymes for DNA replication, they transcribe different forms of RNA. So polymerase 1 makes rRNA, polymerase 2 makes mRNA, and polymerase 3 makes tRNA. So they have different promoter sequences that they would bind to, which allows them to make the proper RNA form. There is a high rate of error in mRNA transcription. The polymerase is not nearly as accurate as the dRNA polymerase. So this creates several mutations, which will add some mutations into our translation discussion. But this also gets into the discussion why RNA viruses are so difficult to create vaccines against, because they're transcribing RNA. They're using that RNA polymerase, which is constantly changing the code, therefore constantly changing the phenotype. So it's hard to get a vaccine that will match a specific phenotype if it's constantly changing. So lots of error in RNA transcription, in just the transcribing of the DNA. The RNA polymerase isn't as accurate. So pre-RNA processing, at first you have this pre-RNA. We want to have mature RNA by the end, right? So we have the words exons and introns. The exons are the pieces that actually code for genes. There's lots of intron in between, right? So we all maybe have the enzyme lactase, right? So our exons will be the same, or else you wouldn't make the same protein. Our introns, this gobbledygook in between, these will be unique between me and you. The introns are what they actually use when they're doing DNA fingerprinting, because the genes that code for, or that code for proteins and enzymes would be the same. So you wouldn't be able to tell if it was me or you who stole the M&Ms. But if you use our introns, the non-coding portions, we would be unique in that place. So we need to cut out these introns, and we need to connect the exons. So you need to know introns are non-coding and exons are coding. I believe that was one of your pre-test questions as well. So how do we do that? We're going to splice them. Splice means to cut. So we're going to cut them apart, and we're going to use what's called a spliceosome. So SNRNAs, a new RNA, the A is missing. SNRNA are going to be used to create these loops. See here? So the SNRNA binds to the beginning and the end of that exon. No, the intron, sorry. And then these will bind together, see here, and that basically creates a SNP. It detaches those nucleotides from the main strand. And so the intron then, I'll fix these letters. The intron is able to be released. So then all that's left is the exons. And again, you have little animations you can look at for that. The final processing, then we need to add protective components to the end. We have a 5' cap and a 3' end, right? Remember the directionality, the 5' near the phosphate. We're going to add a segment of sequence that is recognized by the rRNA. So this is what's going to allow it to attach at the right spot. On the 3' end, you're going to add a poly A tail, many adenine, like junk DNA, basically. And if your DNA is attacked by an enzyme within your cytoplasm, it's less likely to interfere with the genes itself, right? So we're going to splice out the introns. We're going to add a 5' cap that's recognized by recognized by the ribosome, and a poly A tail for protection. All right. So there's some links in there. I'll try to make sure those links are working. It gives you animations so that you can better understand that. So we're headed to the ribosome next, and that's where we will talk about translation. But I'm going to save that for Wednesday because we've gone long enough. I do want you to do the translation POGL. Those are usually pretty easy to make sense of because they give you either information in a paragraph or making sense of the pictures that they provide. Okay, so transcription translation today, and we'll hit, transcription today, we'll hit translation Wednesday. I added in Kelly notes. Those are like cliff notes, I think, for each of the sections we do. This one is gene to proteins. And I've also added in 4 different videos. 6.4 deals with transcription, 6.5 deals with translation. So you can watch those 2 transcription ones over the next couple of days. You can read the segments at the front, you know, and it'll tell you what it's covering. So some of them you might feel more attracted to others because you're like, I have no idea what mRNA processing is. And so you'll click on that probably. I want the translation POGL done today. I think you read 12.5 in order to fill in the notebook. If you didn't read 12.5, you're going to want to read that, you know, sometime tomorrow or Wednesday if you haven't read that one already. Okay, what other questions can I answer?